Hello, I'm Sohini Nath. I'm a junior at UMass Lowell, and this is my research project connection between gene expression and drug targets for an applied drug on a cell line. So my goal of the project is to see if there is a connection between the values of gene expression when a drug is applied to a cell line and what targets the drug hits in the cell line. So there are two databases that I will be using, or rather data sets. The first one is called the links dataset. Now this is a collection of cell lines that are extracted from a variety of human donors. Each cell line has a measurement of its gene expression when a drug is applied to it, as well as the dosage of that drug and how long the drug was applied for. The idea behind the links dataset is to use it to find connections between drugs and the gene expressions on one cell line when it's used. So now I'm going to talk about what is gene expression. When drugs are administered to the human body, they hit targets in the cell that cause the effects of that drug. In the context of links, gene expression is the observation of how turned on or off genes are in a cell, and we can demonstrate this with numerical values. For example, given a donor cell line, without any kind of drug applied to it, this would be my cell line and the values. Now, if I have drug A, the values will change, and there'll be different values. With drug B, you could also see different values. If drug C is applied and all the genes have the same value as drug B, then I can infer that B and C may hit similar targets in the cell, and thus have a connection. The second database that I will use is called TTD, or the Therapeutics Target Database, which is a database for cell targets and the drugs that are directed at those targets when they are applied. So here is the diagram. I have a cell with, for example, 5 targets, A through E. I can see that drug 1 affects A, C, and B, drug 2 affects C, B, and D, and so on. So my research is attempting to combine information from the two databases to see how a particular drug affects a cell's gene expression values and what cell targets are targeted by that same drug. So I identified 3 things that the two databases have in common, which I will be cross-referencing to create one file where I have both information from the links database, aka all the cell signatures, and the TTD database, aka all the cell targets, and combine them to conduct analysis. So what I expect to find once I'm complete with my analysis is that a high correlation coefficient between the two gene expressions for a pair of 2 drugs on the same cell line, meaning similar gene expressions for drug A and drug B on the same cell line, is related to how similar the drugs are with respect to the targets, aka I will see that similar targets the drug hits are related to similar gene expression values. Thank you so much for watching.